This study introduces a novel method for non-invasively recording electroencephalograms, or EEGs, and evoked potentials in awake horses, using a 12-channel active electrode array. The primary goal of this research was to develop a completely non-invasive technique to record cortical evoked potentials in unanesthetized horses, focusing particularly on auditory processing. The study also aimed to characterize the morphology and scalp topography of cortical auditory evoked potentials, or CAEPs, in horses and to identify components homologous to those observed in human CAEPs. The method involves using specially designed brush-shaped active electrodes to facilitate stable signal acquisition through the hair coat, and a multi-electrode array for greater scalp coverage. The innovative method and the findings of the study offer significant potential for advancing the understanding of the equine mind, assessing cognitive abilities, and evaluating normal and abnormal brain function. The study addresses the challenge of recording brain activity in horses, which has been limited due to the difficulty of obtaining stable electrical signals through their dense hair coat, especially in awake animals. Previous studies have recorded EEGs and evoked potentials in horses, but most of them have been conducted under anesthesia or with invasive techniques. The lack of non-invasive methods has been a common problem for other large mammals as well. The brush-shaped electrodes used in this study allow for stable recording without the need for hair removal or skin abrasion, which are usually necessary for using traditional disc electrodes. The active electrodes incorporate small preamplifiers to reduce noise, even when the electrode skin impedance is high. This study is the first to apply active electrodes to equine EEG recordings. The findings provide a foundation for understanding auditory processing in horses and could be applied to other medium to large-sized animals. The study used 3 equine subjects: horse A, a 10-year-old thoroughbred gelding; horse B, a 23-year-old Haflinger gelding; and horse C, a 28-year-old crossbred mare. The horses were presented with pure tones of 1200 and 1500 Hz using an oddball paradigm to elicit auditory evoked potentials. The auditory stimuli were delivered through a powered loudspeaker positioned approximately 1.5 meters from the subject's head. The tones had a duration of 300 milliseconds with random interstimulus intervals varying between 700 and 900 milliseconds. The standard stimulus was presented in 80% of trials, while the deviant stimulus occurred in the remaining 20%. The EEG recordings were performed during daytime in the horses' stables. The horses remained alert and calm during the recordings. The EEG electrode array consisted of 12 active electrodes and 1 passive electrode, all attached to a vinyl sheet cut to fit the horse's head. The active electrodes had silver pins to penetrate the hair, and were connected to a battery-powered unit fixed to the horse's head collar. The central electrode, labeled CZ, was positioned at the intersection of 2 diagonals, each connecting the inner corner of one eye to the medial edge of the contralateral earlobe. The reference electrode was placed slightly below the level of the eyes and a ground electrode was also placed on the head. The electrode positions were labeled according to the International 1010 system, although the landmarks had to be approximated because of the difficulty in identifying specific anatomical points on the horse's head. A figure in the study depicts a brush-shaped active electrode with pins penetrating the hair coat. It also shows the electrode layout with the labels G for ground and ref for reference, and how the electrode cap fits on a horse's head. The EEGs were recorded using a bandpass filter between 0.16 and 100 Hz and digitized at 5 kHz for horse A, with subsequent resampling to 1 kHz, and at 1 kHz for horses B and C. The continuous EEG data were analyzed with EGLAB software and its ERPLab extension. The data were bandpass filtered between 2 and 40 Hz, segmented, corrected for baseline, and screened for artifacts. Segments with artifacts exceeding plus or minus 100 microvolts were rejected. The remaining artifact-free segments were averaged to obtain the cortical auditory evoked potentials. The study reports the number of non-rejected data segments was 15,971 for horse A, 9,871 for horse B, and 1,953 for horse C. The results section of the study begins with an observation of noise-free EEGs recorded from alert horses, with common artifacts such as eye movements seen at the FC1 and FC2 electrodes and subtle muscle tensions at posterior electrodes such as PZ, CP1, CP2, P3, and P4. gross movements were easily detected and those segments were removed from the analysis. Figure 2 shows an example of bandpass filtered continuous EEG of horse B with typical artifacts caused by eye movement and muscle tension. Clear cortical auditory evoked potentials were elicited by both the onset and offset of sounds. The study refers to the CAEPs as onset CAEP and offset CAEP, respectively. The overall features of waveform morphology and scalp topography were similar across subjects. The onset CAEPs consisted of a series of positive-negative waves, labeled EQP1, EQN1, EQP2, and EQN2, that are analogous to the human P1, N1, P2, and N2 components. The peak latencies for these waves were approximately 30 milliseconds for EQP1, 40 milliseconds for EQN1, 50 to 80 milliseconds for EQP2, and 70 to 100 milliseconds for EQN2. Table 1 in the study provides a detailed list of peak latencies and amplitudes of the CAEP components in each of the 3 horses. The offset CAEPs had smaller amplitudes than the onset CAEPs and also exhibited 4 components. The CAEP waveforms were broadly distributed over the scalp. The equine CAEPs had maximal amplitude over centroparietal electrode sites, in contrast to human CAEPs which are greatest over frontocentral sites. The study suggests that the difference in topography may be due to the differing orientation of the sylvian fissure in horses and humans. In humans, the sylvian fissure is roughly horizontal, causing electric dipoles in the auditory cortex to project to the frontocentral scalp. In horses, the sylvian fissure is more vertically oriented, causing dipoles to project posteriorly. The study acknowledges that the location of the auditory cortex in horses has not been confirmed. The left-right asymmetry of the CAEPs differed between horses. Horse A showed largely symmetrical amplitudes, horse B had a slight right dominance, and horse C displayed a slight left dominance. Horse C had the largest CAEP amplitudes, which were approximately 10 times greater than the other 2 horses. Figure 3 shows CAEPs recorded in 3 horses at electrode CP1, and also the waveforms for all electrodes. The figure shows that onset and offset of sounds elicited responses within a 100 milliseconds latency, and over distributed scalp areas. The bar on the time axis indicates the 300 milliseconds duration of the sound stimulus. Figure 4 illustrates the hypothesized position and orientation of EEG sources in the auditory cortex in humans and horses. The discussion section highlights the use of brush-shaped active electrodes to enable non-invasive multi-channel recording of EEGs and CAEPs in unanesthetized horses. This method requires no skin preparation, making electrode application easier and more efficient than conventional passive electrodes. The study emphasizes the time efficiency of the method, requiring less than 30 minutes to prepare a horse, even with a 12-electrode array. This method is an advantage compared to previous methods that used passive electrodes and hair shaving. The new method enhances veterinary care, promotes research, and improves animal welfare by enabling the assessment of brain functions and disorders in awake horses. The study points out that this is the first study to record CAEPs in horses, whether awake or sedated. The recorded responses were similar to human CAEPs and components homologous to human P1, N1, P2, and N2 components were identified. Although the absolute latencies were shorter than in humans, this finding is consistent with the fact that humans have the longest latencies for these components compared to other primate species. The study also suggests that further research is needed to determine if the potential CAEP components in horses represent true functional homologs to human counterparts. The method used for determining the position of the vertex electrode is the intersection of diagonals connecting the inner corner of one eye to the medial edge of the contralateral earlobe, which is described as a convenient way to find the CZ electrode in horses. The researchers acknowledge that this position may not be the exact equivalent to the CZ position in human EEG, but that it is difficult to define referential landmarks for the 10 to 20 system on the horse head. The location of the other non-vertex electrodes is an issue to be addressed in future research. In conclusion, the study describes a new method for non-invasively recording multi-channel EEG and evoked potentials from awake horses. The identification of potential equine homologs to human CAEPs demonstrates the effectiveness of the method for investigating brain functions in horses. This work provides a foundation for further research into cognitive abilities, brain disorders, and the mind of the horse. From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.